Hello and good afternoon and welcome to this Twist Bioscience webinar, Nature and Nurture for Improved Enzyme Activity. My name is Chris Thorne and I am Senior Manager for Field Marketing here at Twist. And I welcome you all today from wherever you're joining us. Before we begin today's webinar, I just want to give you a few housekeeping reminders. Here's a webinar, so you are of course all muted, and that will be the case during the webinar. But if you do have questions, please put them in the Q&A box at the bottom of your Zoom bar, and we will have some time at the end for a question and answer session. Following the webinar, if you have a minute, please do take our brief survey, which just allows us to gather some feedback on the webinar itself, learn why you joined us and whether you got out of what you were hoping to learn. So please do take a moment there. And so we have three speakers today joining us from Protéus by Seqens, and who will cover various aspects of today's talk. First up will be Ronan Rogler, who is Business Development Manager, R&D Services at Protéus by Seqens. Ronan has a degree in chemistry from ENS Paris and an MBA from the Collège des Ingénieurs in Paris. Prior to joining Seqens, in 2019, he also worked at Total and at Global Bioenergies. Following Ronan will be Céline Adignac, who is a graduate from the University Paul Sabatier in Toulouse and has worked with Proteus since the beginning in 1998. And so after 20 years as product manager in molecular biology, she works now as bioservices manager in charge of coordinating customers' projects with the lab teams. And finally, Christoph Ullmann will follow Céline. Christoph studied biotechnology in Strasbourg and joined Proteus not long after Céline in 2000. And he was involved in almost all directed evolution programs performed at Proteus as a technician, assistant engineer, and now project leader since 2012. So with those introductions done, I'm going to hand over to Ronan to introduce the company briefly, and then we'll move on to Céline and Christoph. So Ronan, if I could ask you to turn your webcam and microphone back on, the stage is yours.
Yeah, so thank you very much, Chris. Thank you for the introduction. And we would like also to thank Twist Bioscience for organizing this webinar. So our presentation today is entitled Nature and Nurture for Improved Enzyme Activity. And so we have also a subtitle, which is actually, well, in detail, we'll speak about screening of diversity of native enzymes and directed evolution in order to improve enzyme activity on target reactions. So first, let's start with a presentation of a group Seqens that not all of you may know. So Seqens is specialized in chemistry. So we are a global leader in pharmaceutical synthesis. So we manufacture the active ingredients and also specialty chemicals. So we have more than 3,000 employees. We have 24 manufacturing sites all over the world. And we have also 3 R&D centers. One is located close to Paris, specialized in chemical synthesis, using especially standard chemistry for pharmaceuticals and fine chemicals. We have also R&D center close to Boston, specialized in pharmaceuticals. And the last one is actually in Nîmes, and it's actually Protéus, which is specialized in the development of biotechnology called processes, especially enzymatic conversion processes. So as an overview for the group, half of our activity is dedicated to pharmaceutical synthesis. So we manufacture a large volume pharmaceutical active ingredients, such as, for example, aspirin. We are the leading producer of aspirin in the world. We also produce other APIs such as paracetamol, and we have a catalog of proprietary products. But we also can accompany our customers. It can be big pharma or biotech for the development of complex molecules they identified during their research. So we help them to design a synthesis route. We manufacture the compounds for the clinical trials, and we can accompany them also to commercialization with chemical synthesis of their target APIs. So we have also the other half of our activity is towards specialty ingredients. So in the non-pharma sector, can be in cosmetics, can be also electronics, and a diversity of applications. So in fact, for example, well, as an example, we can manufacture cosmetic active ingredients within our facilities, and in the same principle, we can accompany our customer to design the synthesis and then up to manufacturing scale. So our main R&D center located in Paris, and centers in the chemical processes, so has more than 100 scientists working there. Actually, see, the company Seqens is well, continuously investing in new technologies for pharmaceuticals and fine chemicals manufacturing. Our last announcement were actually that we achieved the construction of our new potent facility. It was in close to Paris in order to manufacture potent active pharmaceutical ingredients, so ingredients which have a very high activity at very low concentration. And the inauguration of the plant took place in summer, and it was inaugurated by the French President Emmanuel Macron. We are very proud of it, actually. So we are also investing for also for pharmaceuticals and fine chemicals in the development of flow chemistry. So currently we are investing in a new pilot flow chemistry plant in our R&D center close to Paris, which is capable of hosting reaction. Well, a reaction which can be difficult in batch mode or difficult, dangerous. So it allows to have more selectivity, safer reaction for nitration, deazotation, or halogenation. So we can produce, thanks to this new unit, we'll be able to produce pre-commercial batches up to the ton scale. And of course, this is just one step in the development of our flow chemistry capabilities. So now I will present you the activities of our affiliate Protéus, which is specialized in biocatalytic process development. So Protéus was founded 20 years ago, more than 20 years ago, actually, and progressively it was incorporated within the group Seqens. So approximately less than half of our activity is dedicated to help the other affiliates of the group. So we use enzymes in order to improve the chemical synthesis of the target compounds, the compounds, well, complex compounds that are targeted by our customers. So in many cases, we can find very efficient biocatalysts that can enable to shorten, so we use less synthesis steps or a more selective synthesis compared to standard chemical synthesis. So together with the team located in Paris, we identify the best combination of standard chemistry and enzymatic catalysis. So the team is located in Nîmes, in the south of France, and we have 25 employees with a multidisciplinary team. We have, for example, chemists specialized in biocatalysis. We have a team specialized in molecular biology in order to use protein engineering and directed evolution to improve the performances of enzymes. This is the main part of our presentation today. We have also a large collection of strains. We have an expertise in microbiology, and we'll also show examples of how we use it, and the team specialized in fermentation in order to upscale the production of the enzymes for our customers. So what differentiates us is that we are offering to our customer a full service, which starts from the initial lab scale identification of an active enzyme. We can improve the enzyme using, exploring collections or using tools such as directed evolution to improve the performances. And we can accompany our customers up to commercial scales for fine chemicals and also pharmaceuticals. So this is pretty unique in Europe in terms of a combination of capabilities. So what are our key capabilities that we gather to propose our services to our customers? So first one is microbiology. So we have a large collection of strains that were gathered during the 20 years of existence of Protéus. And this was built having in mind the applications for biocatalysis. So in fact, a large share of this collection is extremophile strains that can be extremophile for diverse conditions. It can be temperature, high or low. It can be salinity, acidity, pressure. So this allows to identify new enzymes for a variety of conditions in terms of chemical conversion. So we have more than 1,000 of the strains which were fully sequenced, and we can use bioinformatic tools to screen our collections in order to identify selected enzymes active at given conditions. Can be, for example, a thermophile transaminase and other kind of activities that you would be looking for. So this allows us actually to identify from nature's diversity the best starting enzymes. Also, the second key capability of Proteus is protein engineering. So we have two directed evolution technology, and actually these technologies will be presented by Christophe and Céline during this presentation. And the last key capability that we have is that we are integrated within a group, within a chemical group, with commercial scale manufacturing capabilities. So when we start projects, we have in mind a commercial scale, and we can develop for our customers robust processes that can be implemented toward multi-ton scale. And we have a clear methodology for that. As an example of what we can propose, so this is an example of a product that is produced within the group Seqens and integrated within our catalog. It's D-serine. So it's a non-natural version of the amino acid serine, and we produce it with a very efficient process using an aldolase which was developed at Proteus. Okay, it's a very efficient process actually in very mild condition. You see 40 degrees, neutral pH. We use glycine and formaldehyde, no protecting group needed to get in fact this serine with a very high selectivity, high specificity, and also high productivity. So this enzymatic reaction is performed actually in our manufacturing site of Couterne in Normandy, so in France, and we produce dozens of tons of this product every year. In order to produce the enzyme, the biocatalyst, we produce it using fermenters of 15 cubic meter scale at one of our partner facility located in Europe. And for the chemical process, we just need 5% by mass of the enzyme, and everything is filtered after the reaction. So there is no residue of the enzymes in the final product. I mean, it's under detection limit. So it's a very efficient process that is also, I mean, in terms of cost, the impact of the enzyme in the total production cost of this is a one-step process is only 6% of the cost. And actually, this is something we can. This is also a general remark because usually when we develop enzymatic processes, it can be even for pharma, for fine chemicals, food, or large commodity chemicals. Usually, the cost of the enzyme for one synthesis step is not higher than 10% of the total cost. So such process allows access to other non-natural amino acids, and this is a good example of the capabilities of the group Seqens for biocatalytic processes. So now we'll present you actually our methodology to develop biocatalytic processes that we call Sequenzyme. It's our platform for the development of highly efficient biocatalytic process. So this is the process, the robust process that Proteus by Seqens has developed to improve actually biocatalytic process. So first, at the first step, we We identify what is the reaction. I mean, usually our customer, they know the reaction they are looking for, or we can help them because we have a chemist in the group to design a synthetic synthesis pathway. It can be multi-step, but usually there is a specific reaction which is targeted by our customer. So what we can propose is, well, we have developed some kits, and we'll present that, that allow our customers to easily screen in their lab the potency of a diversity of enzymes in order to start if it first, if it works, if you have a proof of concept. Or we can perform it within Protéus, and in this case, we screen the enzyme of the kits, but also we have a lot of off-the-shelf enzymes within our collection, and also we test some enzyme described in the literature in similar reactions. So when you have the first hit, a first enzyme that has a first level of activity on the target reaction, so what you can do is usually what we prefer to do is to find a better enzyme, and for that you can enlarge with a diversity of new sequences coming, for example, from the exclusive library of sequences of Proteus and its partners. It allows again to find better starting points with and also to generate a better IP position because these sequences can have a very low homology and also a much better activity. Then, if it's needed, if you want to further improve the activity of the enzyme, we can optimize them using our protein engineering tools, our two directed evolution techniques, or semi-rational methods. So these are demonstrated tools that we use in order to get a much more efficient enzymes. And Christophe and Céline will actually present some examples of cases where it gave very high results on the improvement of the reactions. And then we apply our methodology to well, to have a robust reaction. We can use a design of experiments methodology to find the best conditions and also to upscale the biocatalytic process, both for the production of the enzyme, the production of the biocatalyst, and also for the enzymatic conversion, the chemical part of the process. So this is actually, we have 7 enzyme families that we propose as kits. So you can see all of them there. Usually there are 7 to 12 enzymes proposed in the kits, and you can directly order it to us, for example, in our website. And it's a very good way to first screen an initial reaction. And actually, now I leave the floor to my colleague Christophe to explain how we can screen the diversity of our native sequences collection.
Since its creation, Proteus has built a large collection of microorganisms thanks to different partners, which give us access to a great potential for generating new IP rights. This collection has been built in respect with the Nagoya Protocol. It represents a large diversity. For example, in terms of temperature, we have from psychrophile to hyperthermophile strains. In terms of salinity and in terms of optimal pH. Until now, we have sequenced more than 1,000 strains, which give us access to millions of potentially exclusive sequence of enzymes, which can be exploited and screened for a specific target activity. Here we have the example of transaminases coming from 60 strains of the Proteus collection. And in this representation made with a software named Cytoscape, we can underline and highlight the sequences in function of different parameters. Here, for example, the substrate's specificity. So we can choose to screen in different enzyme cluster depending on the activity we want to screen. So here we have an example of a project performed with a client who wanted to start a directed evolution program for a target reaction. And he had already several enzymes as potential starting points. We have proposed to this client to extend the screening to our collection in order to identify potentially better starting points in terms of activity and eventually lower similarity with already patented sequence. On this representation, we can see that the enzyme coming from the client selection are close one from each other and far from the selection coming from the Proteus collection. In this case, we have selected 50 sequences representative of the diversity, and we have ordered the corresponding gene at Twist Bioscience as cloned genes in order to evaluate them compared to the already used enzyme by the clients. As a result, we have identified 10 enzymes among these 50 new enzymes that had an activity on the target reaction, which was similar or even higher than the best candidate coming from the client selection. And the identity compared to this enzyme was also very weak. In this table, you can see that the best enzyme coming from the client selection had 50% substrate conversion and was 100% identical to an already patented sequence. And you can also see that the sequences coming from Proteus screening had higher substrate conversion, and one of them and only 50% identity with an already patented sequence. So as a result of this program, we have decided to perform and to start the directed evolution program with an enzyme coming from the Proteus collection and coming from this screening. One way to perform directed evolution at Proteus is named evocyte, and it consists of random mutagenesis. In this case, the starting point is only one gene, and it consists of performing several error-prone conditions using the evocyte algorithm in order to have a library with from 2 to 10 mutations per gene, for example. After that, these error-prone PCR products are cloned, and a sample of each library is sequenced in order to validate the mutation load. And after that, each library is assayed in order to determine the active fraction of each library, which allow us to determine with the evocyte algorithm what we call the optimal mutation load, which is the library where we have the most chance to find improved variants. And this library will be screened to identify the improved variant compared to the wild-type enzymes. This is a way to identify important mutation and position, which can after that be combined by performing, for example, a combinatory library, which can be also ordered at Twist Bioscience. Here we have an example of a directed evolution program performed with company Givaudan. The aim of this program was to produce ambrox from a substrate named homofarnesol, and this reaction is performed with an enzyme called squalene open cyclase. And so after one round of directed evolution using evocyte and random mutagenesis on these enzymes, we have identified 3 variants which have an activity improvement by a factor of 1.6 to 3.5. And after another optimization by design of the experiments of the reaction condition for the best variants, we have another factor improvement of 10. This process is now used at commercial scale by Givaudan for the production of Ambrofix. And you have here the reference of the article describing this work and an extract of this article where we can see the difference of conversion of the best performing variants coming from this directed evolution program compared to the wild-type enzymes. And now let's talk to Céline.
Hi, everyone. So the second technology we use at Protéus to evolve enzyme, it's the L shuffling. The main difference between L shuffling and evocyte is that in this case, the starting point is at least 2 genes which share at least 60% of amino acid identity. The homologous genes to the target one can come from public databases, from a choice of our client, or from our own collection. First, the sequences are optimized to get higher nucleic acid identity and to facilitate the recombination. All the genes are randomly fragmented and reassembled to create new diversity. And in a round of directed evolution, we screen around 10,000 clones to identify variants with improved features like activity or thermostability, for example. Here, it's a work made with Firmenich. The aim of this project is was to improve the synthesis of green notes. Green notes are the fragrance coming from freshly cut grass. The natural pathway can be reconstituted thanks to plant homogenates with soybean for the production of 13 lipoxygenase, the guava for the 13 HPL, and finally, we use the baker's yeast for the isomerization and the reduction of the aldehydes into the corresponding alcohol. In this process, the 13 HPL has been identified as a limiting factor because of its instability and its low concentration in the guava homogenates. So its activity has to be improved to get a commercial process. First, we have worked on the genetic construction of the enzyme to get a measurable activity. Then we have performed 1 round of evocyte and 1 round of L shuffling with the 9 HPL from melon. After that, the best variant has been recombined and new diversity has been introduced with 13 HPL from apple, orange, and tobacco. And 2 other more rounds of L shuffling have been performed. So we have screened 65,000 variants for these 4 rounds of directed evolution. And finally, we have identified a variant with an improvement factor by a factor 15. And furthermore, we have also noticed that its thermostability has been improved by a factor 48 at 50 degrees. And now this process is used also at commercial scale by Firmenich. Here, it's another example of L shuffling. We have worked on an improvement of a cellulolytic cocktail for the production of ethanol 2G. We have worked first on the beta-glucosidase because she was identified as a limiting factor. We have performed 2 rounds of L shuffling on this enzyme. After the first round, the best variant exhibits an improvement factor on 24, and we get another factor improvement of 10 after the second round. So finally, the best variant, the activity of the best variant was improved by the factor 242. After that, we have also worked on the other enzymes of this cocktail, cellulose and endoglucanases, because the beta-glucosidase was no longer limiting. Thank you for your attention.